more importantly the research scholars, students, friends, paper and electronic media and other invitees assembled here. YOGI VEMANA University, I welcome all of you for this two-day national seminar to be organized by Department of Zoology on the theme of animals for human disease, ethical issues and alternative animal models. I feel that this is the need of the hour to conduct this seminar to give some awareness or enlightenment about these animal models. Already the speakers have already mentioned that no experimental work will be completed without using the animal models, particularly in the medical sciences, animal sciences. But the problem is, how long we have been using these animals for our experimentation? As our principal already said that almost 60% of the animal clinical trials are going to waste. And there is a lot of global demand to supply these animal models for the experimental research, particularly in health sciences, medical sciences and animal sciences. We are all aware that earlier days, or those are the days, where even the 10th class people or the intermediate people used to dissect maybe the frog and also the scorpions and also other reptiles for their practical experience. Now, most of the governments have been completely banned to use these animals at a +2 level because of the scarcity and also other demand or to save or conserve these animal models or animals in the nature. It is not surprising that even the plant science people, when they visit the forest, they used to collect a lot of materials, and that leads to some of these endangerment or extinct of the several rare and endangered species. This, because of collections, will destroy the natural ecosystem. Animals are not away from this. Animals also going to become distinct or disappear from the society or from the nature if you are continuing this experimental research using thousands or millions of animals for your experimentation. We agree that so no clinical research will be completed without using the animals, and how to proceed to do research in these areas. I suppose we say that earlier we used to depend on these animal models like mice and rabbits, other things. But there are several models like non-human primates, a group of hominins which are apes and monkeys, sometimes pigs also have been using. Why these animals have been used as a specimen models for conducting this research? That is most important, because they have a greater similarity in terms of physiological and also biological as well as other evolutionary process exactly the same with human beings. In order to test any human diseases, first of all the researchers look at this the animal specimens to test them initially, and they prove that this that is working, the object is working, then this will be applied to the human beings later. But during the experimentation, many results are not encouraging to directly apply or to stress the human beings. That means that research results are going to be waste. So that's why sacrificing the animals or hurting the animals during the experimentation or clinical research are now limited. There are several agencies which postulate the several restrictions or the regulations. One is such institutional animal care and use committee. So institutional animal care, that is the institutional animal ethics committee, in order to conserve or in order to see whether the research is particularly requiring using the animal models for their experimentals, and they suggest whether the animal can be used or not for their experimental work. So like several regulatory agencies throughout the world has been given, but the animals are not able to use it in several experimentation, because they have been continuing these models and giving an alarmic signal about using these animal models in their research. And based upon that several research, it has been shown that there are some alternatives to animal models used in clinical research. Nowadays, for the last 1 or 2 decades, several non-animal testing methods have been comes into picture, and several laboratories throughout the world they have been using this the non-animal testing models like cell-based assays or maybe the microfluid chips tissue models, computer models, as well as the human volunteers. As and when possible, some of the humans can also come forward to test the drug which has been developed and giving very small, minute quantities initially, and to test that whether the drug is really working on the human beings or not. So such type of non-animal testing methods is also now available for your experimental as well as the clinical research. Like human cell and tissue based testing is one of the important one, because it looks like organs on a chip and 3D bioprinting. So this helps and mimics several parameters on par with the humans. Like that, the test drugs will be used using of this animal cell and tissue based testing process. And other things which including tissue bioprinting as well as human volunteer testing, and then computational or mathematical modelings. These are some alternative to animal models for your experimental research. I wish that these today's seminars will directly enlighten us about the issues and challenges of these alternative models for your experimental research. I congratulate the zoology department, more particularly the convener, Professor Venkatraman Reddy Garu, conducting this 2-day seminar and enlightening us about this, the research and alternatives of these animal models. And lastly, the team of the zoology department, all are being actively involving to make this the program as a success one. And they have been inviting the eminent the professors, and they are going to deliver all excellent topics which is related to your theme of this seminar. I congratulate all the organizers once again, and I thank you very much for giving this opportunity. Thank you very much.